Good afternoon and welcome to our Units of Life seminar for this afternoon. I would like to just briefly remind everyone about the Units of Life seminar series and give you a little snapshot of what's to come in the following months before I introduce our speaker for today. And so Units of Life is a seminar series that was born out of the idea that we should try to enhance our interests and community building in the area of cell biology, mechanistic biology here at the Broad. It's been a terrific past year where we had our first inaugural Units of Life seminar series and now starting in our second season. And so the idea is to, again, bring speakers from all over with an interest in mechanistic biology so that we can eventually bridge a lot of those incredible discoveries from genomics and other omics technologies to deep mechanistic biology that can ultimately inform therapies and benefits for humanity. So without further ado, I wanted to now welcome Susan Shao to our Units of Life seminar. Susan is an associate professor in the Department of Cell Biology at HMS. Her lab has an intense interest in a fundamental biological problem, which is really how do you control the quality of proteins during biosynthesis. Of course, there are many errors that can occur. She has a mix of technologies that she uses to answer these questions, both structural biology approaches as well as cell biological and other technologies, which we will hear about today. Susan did her PhD at Hopkins and then did postdoctoral work with Manu Hedge at MRC laboratory in the UK before she was brought to HMS to start her lab. And she's really had a very successful series of new discoveries, I think some of which we will have the benefit of hearing about today. Again, focused on protein quality control and biosynthesis with a focus on the ribosome and what happens to proteins as they exit that and how they get to the appropriate places in various organelles inside the cell. So Susan, it's really tremendous that we have you here today. This is very interesting biology and we can't wait to learn more about it. So thank you so much for joining us.
Thank you, Anna, for that wonderful introduction and for the invitation to be here. I'm really excited to share some of our work with you today. So my lab is really interested in ribosomes and nascent protein biosynthesis especially. And so the two stories that I would really like to share with you today is centered around the ribosomes. And I think one thing to start out, because this is a cell biology seminar series, is to just remind everybody that ribosomes are really abundant because, of course, they're really important. So an average cell has about 10 million or more ribosomes, and each ribosome is generally synthesizing one new protein every minute to 2 minutes. And so, of course, the ribosome has to accurately translate mRNA to make the correct polypeptide sequence, but that's not the end of the story. There's a lot more going on. So after proteins are synthesized, they also have to assemble into complexes, of course, fold, be modified, be localized to the correct part of the cell. And for membrane proteins, they also have this problem of tripogenesis, which is, we think, separate from folding. But again, today, I would really like to focus on the general aspect of what the ribosome does itself. So just as a reminder, ribosomes are composed of two subunits, a large subunit and a small subunit that assemble together on mRNAs at a start codon. And then this ADS ribosome then starts translating through the open reading frame to make this elongating nascent protein until it reaches a stop codon where the release factors will come in and release the proteins that it can go off, be folded, and do whatever it needs to do. Now, every so often, something bad happens. And so, for example, you may have mRNAs that are generated from mutated DNA that are damaged or that are misprocessed. And any of these defects can cause ribosomes to stall during elongation. And when this happens, of course, if the ribosome stalls permanently, then the ribosome is trapped here with a nascent protein that is incompletely synthesized. And this protein, if the ribosome doesn't recontinue, especially in this case where the mRNA is basically cut off without a stop codon, then that protein is never going to reach a native protein state, right? And so, of course, if we let these proteins, incompletely synthesized proteins, persist in the cell, then they tend to be proteotoxic. aggregate, then you have dominant negative effects. And this often induces pathological conditions such as neurodegeneration or developmental disorders. And so, of course, to avoid these, the cell has numerous responses to try to prevent all of these bad conditions from happening. And so we know that stalled ribosomes can activate quality control processes, including mRNA degradation to get rid of the faulty mRNA that often causes ribosome stalling, to degrade this incompletely synthesized nascent protein, and then various cellular stress responses to try to save the cell from these proteotoxic effects. And so one central question that my lab and a lot of other labs in the field have been interested in is trying to figure out how quality control factors can identify these very rare aberrant instances of ribosome stalling or aberrant ribosomal complexes when you're in this very abundant cytosol with millions and billions of ribosomes. And specifically because ribosomes are so big, a ribosome that's in trouble often looks very similar to a ribosome that's still working, right? And so what are the actual cues, molecular cues that allow the few quality control factors find these very rare aberrant ribosomes? And so this is a very bustling field, so I won't have time to kind of go into everything that we know. I'll just say very quickly that we now know that these ribosome collisions, when one ribosome runs to another, is kind of a very common signal that activates protein quality control. But one thing that I would like to talk about of research that we recently did is to talk about what happens after stalled ribosome is initially recognized. And after the stalled ribosome is recognized, usually it will recruit factors to split that ribosome back into the large subunit and the small subunit. And specifically, there are two sets of factors that can do the splitting depending on how the ribosome initially stalls. And so when you do this on a translationally stalled ribosome, what happens is you'll release the small subunit with the mRNA, and this will usually lead to mRNA surveillance pathway. So we'll degrade that mRNA, again, getting rid of the substrate that often causes ribosome stalling. And the other thing that will happen is that if you have this translating ribosome that gets split, is that you'll have the large ribosomal subunit. But often in this case, it will be stuck here with the nascent protein still trapped on the large ribosomal subunit because there's a bulky 26 kilodalton tRNA on one side of the subunit. And if this protein has any bit of a folded domain on the other side of the ribosomal exit tunnel, that will basically kind of keep it on the large ribosomal subunit. And it is this very unique species where the large ribosomal subunit can be distinguished from empty large ribosomal subunits that then activates a so-called the ribosome-associated quality control pathway, or RQC pathway, that eventually degrades this nascent protein. And this is a pathway that we've been very interested in for a long time. So the general theme of the two stories I'd like to talk about today is actually not focused on the protein or the mRNA, but actually focused on what happens to other components on these ribosomal complexes. And to do that, I want to first go dig a little bit deeper into this protein quality control pathway because it actually is linked to another surveillance pathway that we recently studied. And so RQC, of course, is not just a RQC, but it actually requires proteins to come in and activate this protein degradation. And so these proteins include a factor that selectively recognizes the interface of the tRNA still trapped on the ribosome, which then recruits an E3 ligase that helps mediate polyubiquitination of the nascent protein on the ribosome. It does this P48 link ubiquitination, which is usually the signal for eventual proteasomal degradation. And this ligase is called lusterin. And so our contribution to this pathway previously was to reconstitute this pathway in a cell-free system, both with lysates and with purified factors. And so one key that we took advantage of was to use an in vitro translation system that you can program with any mRNA of your choice. And so we chose to program it with a truncated mRNA that lacks a stop codon. So therefore the ribosomes will run to the end, have nowhere else to go. And that basically reconstitutes the stalling pathway. And so if we do this, we can then also label the protein that we're synthesizing, which is shown here. And we can show that this protein is still trapped and attached to tRNA, this band here, by adding RNAs to show that that can collapse it down to the native side. So that basically says that we can remake this complex here and selectively follow the fate of the nascent protein in this system. We can then purify those stalled ribosome complexes and incubate it with all the known factors that we had, the field had identified before, to then reconstitute aspects of this reaction. And so specifically in this case, we can track the polyubiquitination of the protein, which you can see here with the signal starts migrating up through. It's very possessive polyubiquitination, only when we add in all of the factors needed to split the ribosome and the two factors that are that rely on RTC. And the last thing that we can then do with this cell-free system is, of course, then isolate these complexes for structural analysis. And so this is really the basis of the cartoons that I've been showing here, where we can actually see these different factors, how this NEMF factor is able to selectively recognize this interface down to the tRNA, and then also help recruit that E3 ligase here, which wraps all the way around the large ribosomal subunit so that its catalytic ring domain is placed right outside the exit tunnel, that it can modify the proteins that are coming out of this ribosomal exit. So that's kind of where we were right around the time that I started my lab. And also around that time, there was another factor in this pathway that was identified called ANKZF1 in humans. And it was shown that ANKZF1 acts in the RTC pathway downstream of ubiquitination. And its specific function was to release the nascent protein from these trapped large ribosomal subunits so that they're actually free to be degraded by the proteasomes. And so we can add ANKZF1 into our reconstituted reactions here. And if you just pay attention to this last row, which is to show that if we reconstitute the entire reaction and then add in this ANKZF1 protein, now we see this band down here. We omitted listerin in this case so that we don't have the polyubiquitination, so we can really trap this reaction. But we can also show that if we did include listerin, the ubiquitination of protein has no effect on whether or not ANKZF1 can release this protein. So again, this is very much like RNase before. It's basically separating the protein from the tRNA, which is what we're seeing here. Now, ANKZF1 is a quite interesting protein, specifically because it has this domain that looks like a translation termination factor. And ERF1, the factor that it has some homology to, its role is also to release proteins after translation terminates by hydrolyzing this ester bond between the nascent protein and tRNAs. And I'll remind you again that all tRNAs end with this 3' CCA nucleotide sequence. And so based on this homology, it was then hypothesized that ANKZF1 would do the thing. It would basically cut this bond here and then let the nascent protein go. But if you look at the gels that we had seen very carefully, you might notice something a little bit weird, which is that if we run the reaction with ANKZF1, it produces a band down here showing that the protein is being separated from the tRNA, but it's actually just slightly larger than the band that is produced if you add in RNAs. And also, then if you take this reaction where you have generated nascent protein band, and then add RNAs, you can then collapse it down. And so what that means is that actually ANKZF1 is not hydrolyzing this ester bond. What's actually doing is, we predicted, is that it is generating a protein that is still attached to a little bit of tRNA here. And we hypothesize that it's part of the CCA sequence. And so if it's actually, instead of hydrolyzing this ester bond, it's cleaving the tRNA, that suggests that there's another part of tRNA that's also cleaved and non-functional, this 5' tRNA fragment. And so one of the first projects in my lab was conducted by a student, Matt Yip. And the main question that he asked was, what happens to this 5' tRNA fragment? And so to do this, he again went to an in vitro system. And instead of radiolabeling nascent proteins in a translation system like we normally do, he added in radiolabeled tRNAs. And the way that he did this was really cool. So he could either incorporate the radiolabel on all of the C's so that he could assay the CCA here, or on all of the U's where it would not be included in that CCA sequence. And so he can incorporate these tRNAs into these translation reactions. And again, they are incorporated into this protein tRNA adduct. And if he then adds in that ANKZF1 protein, or the yeast homologue in this case, you'll see that in both cases, the wild type protein induces a cleavage of the tRNA, which we can assay here. but only the C-labeled tRNA has this additional band that corresponds to the same band as that nascent chain. And so that suggests that a part of the CCA is still attached to nascent protein, but this U down here, for example, is not. And then again, because this is a catalytic enzyme, we can make a mutation and show that that enzyme is not functional for cleaving these tRNAs. And so that's just shown here. Those are the two cleaved, but only the C has that additional band. We can then also run this on a separate type of gel to get higher resolution to see this tRNA cleavage down here. Okay. So then I'm going to summarize a lot of work that Matt did and basically say that he can then map the cleavage site and show that this ANKZF1 or the use homolog VMS1 specifically cleaves off the CCA of the tRNAs that are attached to these nascent proteins. And by doing so, this has two implications. The first is that because all tRNAs have CCA, so tRNAs generally have very different sequences, but they all share that CCA. It means that this protein can cleave peptidyl tRNAs on ribosomes that are stalled at any single codon. So it's a very generalizable mechanism. The second thing is that the way that VMS works as a nuclease actually generates a unique intermediate. So of course, this is a 5' fragment. It cannot work in translation. And it also has this 3' chemistry called a 2', 3' cyclic phosphate that is at the very end of this tRNA. And so again, these tRNAs cannot function in translation. The question is what actually happens to them. And to do this, we had to learn a lot more about tRNA biology. We generally work with proteins, but luckily there's been a lot of work done on this. And we learned that the 3' CCA nucleotides are not genomically encoded into tRNAs, they're added on post-transcriptionally. And actually tRNAs are generally transcribed as precursors with these 5' leaders and 3' trailers that are then cleaved off by different nucleases to generate this intermediate. And a single protein, tRNT1, adds on the CCA nucleotides onto these all tRNAs. And this happens in both the cytosol and the mitochondria. And what's really cool about tRNT1 and work in Philip Sharp's lab is that it also has this kind of quality control ability where if the tRNA is not well folded in this going and add on more nucleotides, and that is the signal for tRNA degradation. And so because of this kind of proofreading activity of tRNT1, we were quite interested to see if tRNT1 can act on the tRNAs that we found were cleaved on stalled ribosomes. And to do this, again, we turned to an in vitro system. We set up reactions where we made the cleaved tRNAs, and then we added in tRNT1. And what we see in showing here, so if we don't have the cleaving factor, nothing happens. If we have the cleaving factors on these ribosomes, we get cleavage. If we just add in tRNT1, actually nothing happens. But then if we add in T4PNK, which gets rid of that 2' cyclic phosphate, now we get all full-length tRNA. Now, because we did this all in a single tube, we can do order of additions, but in this case, we did a single tube. At this time, we couldn't tell if tRNT1 is adding back these CCAs or if they're preventing cleavage, right? But one way that we can show that is actually preventing cleavage is by the following experiment, where we do the same reaction with unlabeled tRNAs and only add in the radial label at the very end on the CPPs. And so tRNT1 uses CPPs to add in the CCA, and only the reaction that has all of these factors are radial label, showing that it's cleaved first, and then only the radial label CTPs can be added on right there. Okay, so as a quick summary, I hope I've shown you so far is that these stalled ribosomes are split by factors that generates this very unique 60S large ribosomal subunit complex that is selectively recognized by RPC factors. One of these RPC factors, ANKZF1 or VMS1, is a protein that cleaves the peptidyl tRNA to release this unique protein attached to a little bit of tRNA for proteasomal degradation, and this tRNA intermediate that then has to be recycled. And this recycling occurs through tRNT1, and it requires a dephosphorylation event. We used an enzyme from a bacteriophage to get this dephosphorylation to happen. And so the mammalian one is still not known, but I'll tell you a little bit about that next. And then tRNT1 can then add on these CCAs again to regenerate tRNAs for translation. And what we really like about this is because tRNT1 also has this proofreading activity, it suggests that these tRNAs are obligately checked for their integrity. during this RQC process. And so what's kind of nice about that is, of course, what the initial cause of stalling is not known. So maybe that the tRNA actually is the cause that initially caused the ribosome to stall. Okay, so what about this enzyme right here? So we had put this together. This was really bugging us. And so Matt decided to go after it. And he did this in a really clever way. So he took advantage of a self-cleaving ribozyme that also generates the exact same chemistry on the 3' end. And then he transcribed these RNAs with this 2', 3' cyclic phosphate, added in tRNT1, and then took various lysate fractions to try to track the activity, because we know that the CCA addition requires this 2', 3' cyclic phosphate removal. And so shown here is just, a fractionation of lysate, where you can see that in each fraction, there are different combinations of proteins, but only in these fractions is there a special protein, perhaps, that is mediating this 2', 3' cyclic phosphate removal. So we did this through several steps and then sent all of the fractions for mass spec. And in the end, we came out with a protein called ELAC1. And so ELAC1 is a homolog of RNase Z, which is why it kind of stood out to us, because RNase Z catalyzes this last step of 3' tRNA trailer removal to generate the precursor for CCA addition. And so ELAC1 is present in all kingdoms of life, but not in all eukaryotes. So bacteria have ELAC1, but for example, S. cerevisiae has lost ELAC1 during evolution. And ELAC1 is a dimer in cytosol. In addition, there's another homolog, ELAC2, and this is actually the protein that does all of this 3' trailer processing in eukaryotes. So it does this in the cytosol, and it has a mitochondrial targeting signal that allows us to do it in the mitochondria as well. But ELAC1 was the only one that came out of our screen, and then Matt tested it for its activity. And shown here is just his tRNA substrate that is radio-labeled, that he can incubate with wild type or a catalytic version of ELAC1. And you can see already that there is a slight shift in the tRNA here with wild type and not mutant ELAC1 that suggests that some of the chemistry is changing. And then he can add in tRNT1 and show that the wild type but not mutant ELAC1 is just as good as the bacteriophage enzyme T4PNK and mediating CCA addition by tRNT1. And it can do it at much more substoichiometric amounts. I'm not going to show the data, but this also works on VMS1 cleaved tRNAs that we can generate from ribosomes. And so the next thing that we wanted to see is that we can show that ELAC1 is sufficient for this activity, but is it necessary? And to test that, we generated ELAC1 knockout cells. So the first thing that we did was to see how if we incubated wild type lysates with our tRNAs, you get this kind of very heterogeneous modification. If you knock down tRNT1, you get less of it, and you can rescue that by adding back tRNT1. And now to the same system, if you added lysates generated from ELAC1 knockout cells, again, you get this deficient modification that can be rescued by ELAC1, but not a catalytic mutant version of the protein. So again, these are the systems where we can get full rescue of the activity. So this shows that ELAC1 is also necessary for this repair process. And so the next question that we want to ask is, of course, in these cells, we're only knocking out ELAC1. What about ELAC2? And because in the lysates, ELAC2 is still present, and then all lysates are still able to process the tRNAs. We can generate these knockout cells, which means that their tRNAs are just fine. And so Matt then did this experiment where he compared how ELAC1 and ELAC2 can process tRNAs that either have this 2', 3' cycle or that have a trailer mimicking the biosynthetic route. And so if you did this with a 2', 3' cyclic phosphate with ELAC1, you can see that he can get the CCA addition working really well. But if he now uses this delta CCA with ELAC1, you actually see very little amount of CCA addition. Where it's very poor at removing that 2', 3' cyclic phosphate, but it's quite good at removing the CCA trailer and adding the mediating the CCA addition here. This is in mammals. In yeast, of course, they don't have ELAC1, and so they only have a single enzyme called TRD1. And what's interesting when they only have one of these homologs is that it's equally both good at mediating both types of processing. And so what this suggests is that in mammals, these RNase V homologs seem to have specialized, where ELAC1 is specialized for tRNA repair in removing this 2' 3' cyclophosphate, and ELAC2 has specialized for tRNA biogenesis and cleaving off this 3' trailer. And both of them then converge on this intermediate that can be acted on by tRNA1 for the CCA addition. And so as a quick summary at this point, we can put in ELAC1 into this map here, where during RQC, these tRNAs are cleaved and then recycled by this two-step process. So at this point, our lab is really thinking about what's happening to these aberrant ribosomal complexes. And we knew that if ribosomes stall during translation, they will activate mRNA degradation and nascent protein degradation. We can then show that there's this functional link between nascent protein degradation and tRNA recycling that then links to a tRNA quality control process. And so that then led us into a completely new research direction, which was to ask about what actually happens to other components of stalled ribosomal complex or just aberrant ribosomal complex. And specifically, we were quite interested in what actually happens to the ribosome itself. So to the rRNA and the ribosomal protein. And so just as a quick reminder, the ribosome is this amazing machine. It's really large. It has 4 rRNAs and 80 ribosomal proteins that all have to assemble really carefully onto this ribosome. And so all of these proteins are synthesized in the cytosol. Many of them are imported into the nucleus, where the assembly actually occurs in nucleolus. And so eukaryotes, of course, don't have operon. And so there are many instances, for example, where the amount of ribosomal proteins individually may not be highly coordinated in their synthesis. And so these excess so-called orphan ribosomal proteins that fail to assemble into ribosome may be quite toxic for the cell. And one factor in mammals that mediates the degradation of these orphan ribosomal proteins is a protein called UBE2O that can recognize these unassembled ribosomal proteins and then target them for proteasomal degradation. Now, at the same time, the levels of ribosomes themselves also have to be very carefully regulated. And so ribosome levels can influence the repertoire of mRNAs that are actually translated in the cell. And what's really remarkable about UB2O is that it also plays a really important role in regulating ribosome degradation during red blood cell differentiation. And so it can actually, if you overexpress it, drive quantitative ribosome degradation, but its specific role in this case is really in the later stages of red blood cell differentiation, because in this time, you have to upregulate ribosomes to synthesize a lot of hemoglobin, but then actually degrade all the ribosomes to get a bag of hemoglobin that makes up your red blood cells. And so because of these two really important functions, UB2O levels have to be really carefully regulated. It's expressed in all tissues at a low level for this quality control function, but is specifically upregulated during red blood cell differentiation for this ribosome degradation function. And consistent with these roles, knockout of UB2O causes anemia, but it's also amplified in cancers where we think that the levels of ribosomal protein synthesis are more likely to be uncoordinated. And so UB2 is a component of the ubiquitin proteasome system. It's an actually very unique component of the UPS because it proteins in this system of thousands of proteins that has both E2 ubiquitin conjugating activity, meaning that it can have a ubiquitin on a cysteine and then transfer it to a substrate, as well as E3 substrate recognition activity. And so it also has a really unique component where it primarily catalyzes multi-monoubiquitination of lines. Instead of building chains, it really coats proteins with ubiquitin. And so it looks like this here. It's a pretty large protein. This is the catalytic UBC domain that has this catalytic cysteine that has E2 activity. And then previous studies have shown that this N-terminal conserved region 1 and conserved region 2 seem to have this E3 ubiquitin ligase activity that is able to recruit substrates. And so like ribosomal proteins, UB2O clients generally have basic and hydrophobic patches generally, but they're otherwise very diverse. And so unlike a lot of E3 ligases, there was not a specific degron that can fuse onto a protein and show that UB2O can then degrade it. And so we were quite curious about how UB2O is able to recognize ribosomal protein for degradation. And again, this work was carried out again by Matt with help from Sam, who was another graduate student in the lab. How UB2O recognizes clients, we still first needed a model client. And we decided to look back at this process where UB2O is really important. So again, UB2O is upregulated during this late stage of red blood cell differentiation, where it's degrading ribosomes in this transition from reticulocytes to erythrocytes. And also during this stage, reticulocytes are actually the cells where we get our lysase to do our in vitro translation reactions. And so previously we had collected a lot of data, cryo-EM data of ribosomes from these reticulocytes. And we had classified them systematically and identified a protein that seems to be upregulated in reticulocytes and that binds ribosomes. This protein called IFRD2 binds the catalytic center of the ribosome where it mimics a key site tRNA and mRNA. And so by doing this, by binding ribosomes in this way, basically it means that any ribosome that's bound to IFRD2 cannot translate mRNAs because tRNAs can't get into this part. The other thing that was interesting to us about IFRD2 is that if you look at its expression during erythropoiesis, it mimics that of UB2O. And if you look at proteomics data sets of cells, of wild-type cells or UB2O knockout cells, IFRD2 protein levels are elevated in cells that have that are knocked out for UB2O. And so this suggested that IFRD2 may actually be a client of UB2O. So to test this, Matt did a very simple experiment where he simply co-expressed UB2O with IFRD2, and then he pulled on IFRD2 to see if they interacted. When he did this, he saw that in cells that have wild-type UB2O, you can see ubiquitinated IFRD2, suggesting that UB2O is mediating IFRD2 ubiquitination. And this doesn't happen in cells where the catalytic cysteine of UB2O is mutated. But what was more surprising here actually is that when he did this pull down, you can see that the IFRD2 interacts with wild-type UB2O, but not the catalytic dead enzyme. And this was quite surprising because again, we only mutated a single cysteine in UB2O, and that cysteine is in a catalytic domain and not the putative substrate binding domain. And so he got a little bit of insight into what was happening here when he did the reciprocal pull down. So same cell, but now he's pulling on UB2O. And when he did this, again, he saw that IFRD2 only interacts with wild-type UB2O. But again, if you look really closely, the population of IFRD2 that is actually interacting with UB2O is not this unmodified population, but actually these larger molecular weight populations, suggesting that it's the ubiquitinated form of IFRD2 that is selectively interacting with UB2O. And this is why you need that catalytic activity. And so this suggests that UB2O has an annotated ubiquitin binding domain, which you can then test by just fusing ubiquitin onto IFRD2. And so if we do this and put ubiquitin in front of IFRD2, now that unmodified version, which still has the ubiquitin on it, can interact with UB2O, and it can also interact with the catalytically dead version. So suggesting that actually just getting rid of that cysteine doesn't have any effect on substrate binding. And then ubiquitin has a hydrophobic patch, which is shown here, that is often used for binding interfaces. And if you mutate the residues in that hydrophobic patch, we can then abolish that interaction again. This interaction, of course, is important for function. If we use the same things and carry out ubiquitination activity, you can see that UB2O mediates the ubiquitination of ubiquitinated IFRD2 much more processively than the other two versions. And so what that suggests is that UB2O is able to bind clients like IFRD2 transiently, but only if that first ubiquitin is added on does it kind of stick better to UB2O, and that mediates a very processive ubiquitination. And so this basically is a feed forward mechanism where if one ubiquitin is added on, then you get a lot more added on. Now, at the same time, Sam was also studying, basically, she was looking at all 80 ribosomal proteins, and she was trying to see which ribosomal proteins are UB2O clients. And by doing this, in work that I'm not going to show, she basically can see that about 2/3 of ribosomal proteins are ubiquitinated by UB2O. But when she was doing all of these systematic studies, another thing that she realized is that she was doing pull downs, for example, is that she kept on seeing these ribosomal proteins that are UB2O clients interacting with UB2O, but there was another protein that was coming down at like basically equal amounts. And this protein was a protein called NAP1L1. And this is a homolog of yeast NAP1, of which yeast only have one, and yeast NAP1 is best characterized as a histone chaperone. But humans have about 20 different paralogs of NAP1, and they're all assumed to be histone chaperones, but that actually may not be the case. What we can see with NAP1 is that NAP1 and UB2O form a stable complex. And unlike other UB2O clients, NAP1 interacts equally well with both wild type and catalytically dead UB2O. And no matter how hard we try, we never actually see UB2O mediating NAP1L1 ubiquitination. So that's not a client, but it's actually defining UB2O for some other reason. And this is just showing that we can purify a really stoichiometric complex of the two proteins. And so because NAP1 did not seem to be a client, but it still interacted, Sam was really quite interested in whether or not it had a functional effect on how UB2O modifies its clients. And so I have to say that I personally was not very optimistic about this particular route, but she persisted and made a number of knockout lines. So here on this slide is with UB2O knockout by itself, and then she also made double knockouts with NAP1L1. And she's then looking at UB2O interaction with a ribosomal protein that she can show as a UB2O client, UL2. And first, what you can see at the left is that, again, UL2 interacts better with wild-type UB2O than CD. It's also ubiquitinated, showing that it's a UB2O client. And then as she knocked out UB2O, she can basically abolish both the interaction and the ubiquitination of UL2 here, showing that NAP1L1 is very important for the ubiquitination of UL2 by UB2O. You can show that this effect is specific. So again, in these knockout lines, you can then add back, re-express NAP1L1, and then rescue this defect. But if you remember IFRD2, NAP1L1 is not important for every single UB2O client. So IFRD2 does not care whether or not NAP1L1 is there. So to get a little bit more into the mechanism, the other thing that Sam can show is that NAP1L1 can interact with UL2 in the absence of UB2O. These are UB2O knockout lines. And if you re-express UB2O, it doesn't actually affect the interaction between NAP1L1 and the client. But the very best interaction that you get with these factors is when you have all components there. So if you have a ubiquitin on the client, NAP1L1 present, and UB2O present. And so it's really these separate multivalent interactions that we think are all low affinity individually that are actually building up together to basically choose substrates. And so just as a summary here, basically, again, we have these interactions that are possible between ubiquitin and UB2O, ubiquitin with the client and NAP1L1, and NAP1L1 and UB2O. And we really like this model because what it suggests is that you can have these very heterogeneous clients that bind weakly to one part, but if you add up all of these interactions, you can get very specific substrate selection. Okay. So I have, so while Sam was doing this and identifying NAP1L1, this actually helped Matt a lot. So Matt, during this entire time, was trying to solve the structure of UB2O, but he was struggling quite a bit with something that we call preferential orientation, where UB2O, we can only see one view of UB2O by Crowley-Young. But one way to solve preferential orientation is to assemble your protein in a complex, and that can magically help a lot. And so then Matt was able to solve the structure of UB2O in complex with the NAP1L1 dimer shown here in purple. And so NAP1L1 basically forms this platform that then binds to the CR1, CR2 domain of UB2O in blue here, which then connects to this catalytic UBC domain in green. And so this is where the catalytic cysteine is. And we think that the CR1, CR2 kind of platform is basically making an interface where substrates can bind and reach the catalytic domain of UV2O. And so here is just a close-up of where we think clients are binding. Again, they're very heterogeneous, so we think that they're kind of like moving around in this cavity. And there are a few other things that can modulate how they bind, specifically this kind of flexible hairpin loop on NAP1L1 that we can only see at low resolution. We can then show that NAP1L1 binds to UV2O primarily by charge-driven interactions. So NAP1L1, it's a histone chaperone, right? So it's completely red, it's very acidic, and it binds to this basic region in UV2O. And this interaction is not very specific. It's really just charge and shape complementarity. So it takes multiple mutations before we can actually abolish it. And so one other thing that I really like about this is I told you before that there's about 20 NAP1 paralogs. They all kind of have this shape and charge distribution. And we can now show that there are multiple NAP1 homologs that seem to bind to UV2O. And so one speculation for the future, perhaps, is that different NAP1s serve as different kinds of adapters for UV2O. And if you compare the structure of UV2O in complex with NAP1 with that one view that Matt saw with a UV2O without NAP1, these are what we think are analogous views. And if you look at this column with CR1 and CR2 versus where we think the UVC domain is here, you can see that the placement of the UVC domain is quite different in these two complexes. You can also see this in this low resolution map where we try to fit in the model that we generated with the complex, and basically this UVC domain is kind of sticking out. And specifically in the apial structure, it seems like the UVC domain is kind of swinging inward against and clamping against the CR1, CR2. And one implication of that is that the UVC domain may then interfere with substrate binding, which would be happening in this cavity here. And so we can test that by deleting the UVC domain. And if we do that, now we can see that UV2O without the UVC domain can actually bind unmodified IFRD2 even without ubiquitination. So basically, if you take away the UVC domain, those initial contacts are a little bit better. And this suggests that the UVC auto-inhibits the client binding activity. And this is a little bit opposite than most E2, E3 enzymes interactions. So basically, usually the E3 will activate the E2, and the E in the E2 will have no effect on the E3. And so that made us ask what actually happens to the E2 activity. And so basically, the UVC domain of UV2O looks very similar to all other UVC domains. And I told you before that it's pretty well known in the field that if you have an E2 that has this low activity, and this activity is measured by ubiquitin discharge from that cysteine onto free lysines, which happens at a very slow rate. It is actually amplified if you add in a cognate E3 here, RNF4 for UBCH5. That rate is now greatly increased. So this is for a control E2 enzyme UBCH5. If we do the same activity with UV2O, we see something very different. So UV2O basically has a high intrinsic E2 activity that is not affected if we add in the substrate binding part. Okay, so the last thing that I'd like to just share about this entire story is the next thing that we wanted to do with the structure was to really see how UV2O is able to bind ubiquitin. And so Matt was able to assemble a complex again of UV2O NAP1L1 with a ubiquitinated client. And you'll see here that this is the structure there. Basically, everything looks pretty much the same except for this one additional density in orange here that we can very precisely show is ubiquitin. We also had client here, but we don't see client. We think it's actually quite conformationally heterogeneous around here. And then the other thing about where the ubiquitin is placed relative to the active site, it's basically about 30 angstroms away, far away, for the UVC domain to actually modify the ubiquitin itself. And so we think that that's why UV2O is not using this particular site, for example, to build ubiquitin chains onto substrate. And so the nature of this interaction can be shown here. Again, we had already known from our previous biochemistry that this interaction is probably using the hydrophobic path of ubiquitin. And if you see how it interacts with the SH3 domain, there's a corresponding hydrophobic path on the SH3 domain that is mediating this interaction. We can then mutate these two isoleucines and show that indeed that abolishes the ubiquitin-dependent association between UB2 and a ubiquitin IFRD2. And it also, of course, has a functional effect. And I really like this particular experiment where if we use the ubiquitin binding mutant of UB2, we still see monoubiquitination, meaning that that first interaction, that first ubiquitin happens, but we don't see the processive ubiquitination that happens afterwards. So basically it's touch and go, it's no longer touch stick and then add more ubiquitins. And for that same reason, now it doesn't matter if you have a ubiquitin on your client. Basically, both types of clients look exactly the same. You only get that first ubiquitination event and not anything else. And so just to kind of push that home, what we think is happening then is that if you abolish that ubiquitin binding activity, you no longer get this processivity or the speed forward effect. And so in summary, I hope that I convinced you with that other story that UB2 is a really interesting enzyme that has this intrinsically active E2 domain that inhibits client binding, and that this auto inhibition can be released by a chaperone repurposed as the quality control adapter like NAP1L1 that basically binds and potentially relieves this contact that was made intramolecularly. And that client selection then occurs through multivalent interaction that can occur through multiple components in the system. And one of these interactions is mediated by this novel ubiquitin binding domain on UB2O that can basically trap ubiquitinated substrates and mediate this feed forward mechanism. And then kind of taking a step back to the big picture, we're still really quite interested in all of these big questions about what's happening here at the molecular level. And UB2 is just kind of one of the places that we're starting on this particular site here. And so with that, I'd like to thank my lab. Matt and Sam did all of this work. Matt just graduated, so we're glad to see him go. And Simona's in the GE lab helped us with the mass spec to identify ELAC1, and of course, a lot of help from the Cryo-EM Center at HMS. And I'm happy to take any questions.
Any questions online? Here, yeah.
So the stated function of UB2O, right, was the degradation of excess ribosomal protein.
Yeah.
So this model that we laid out, with NAP1L1 and the processivity of ubiquitination, have you tested whether they're specifically required for this degradation of excess? Like what happens if you mess with that in, say, a red blood cell, for example?
Yeah. So we haven't looked in red blood cells, but Sam is following up with the specificity question where we made the NAP1L1 cells and we're doing proteomics. And it seems like in 293T cells, ATK293T cells, NAP1L1 is really important for most ribosomal protein degradation. NAP1L4, for example, if we knock it out, it also interacts with UB2. It doesn't have as big of an effect, and we haven't really found those substrates yet. And so we think that NAP1L1 at least does have a very important role for a large subset of UB2 clients.
More questions? I had a question. A couple of them actually, but this process of avidity gained from multiple interactions. Can you speak to whether that you think is generalizable to other kind of systems where you might have, clients and pockets, and the way that there needs to be some built-in promiscuity, but also needs to be the diversity of possible clients, or maybe other systems in which you have seen this?
Yeah. So I think certainly the idea of multivalency driving client selection is not completely new, right? There are a lot of other systems, for example, the APC that has multivalent interactions that help stick the right clients at the right place, also ubiquitin binding. This, I think, we think it's a little bit special because of how diverse the UB2 clients are. And we do think that that might be a generalizable principle for quality control, especially where you're trying to identify the absence of correctness, where it may not just be one specific wrong thing, right? There may be almost infinite number of things that are wrong. So yeah.
Does make sense. And then the other question that I had, the kind of cofactor NAP1 story that you mentioned, you said that it's required for a subset of clients, right? And so I was wondering, out of the like 80 ribosomal proteins that you need to make, is there any rules in the game of like who are the clients and are there any ways that one could
Yeah.
gain something from that?
Yeah, so I mentioned that Sam had done the screen. About 2/3 of ribosomal proteins seem to be UV2O clients. And if we plot them by PI value, there's kind of like a really nice cutoff where above PI 10.34 or something, most of them are UV2O clients. The other thing that makes proteins more likely to be UV2O clients is how well they aggregate. And so this is where we think like chaperones like NAP1 may play a specific role in basically a lot of other E3 ligases won't be able to capture these proteins before they aggregate. And so maybe you have adapters that are bringing them in.
Other questions from the group? Oh.
So regarding the first story, right? So with like protein like MCF1, you're getting cleavage of CCA, but then you can also regenerate it. I was wondering how much of that process, after you've dissociated the ribosome, how much of it is reversible? Once you've dissociated, are you definitely going to be degraded and your DNA recycled? Or is there a process for which you could reassemble or come back? Yeah.
So once you split the ribosome, it's almost impossible to go back. And the reason for that is because if you had to go back, you basically need that tRNA to find the right mRNA codon again to restart translation. So if you split ribosome, they stick together really well, right? This is a huge interface between the large and the small subunit. And so there are kind of what we call dead end intermediates where the small subunit just kind of sticks back on. But how often that actually results in translation happening again, I would bet almost never, probably. Not impossible, but I would say.
And another question about NAP1L1. Is it actually a histone chaperone in addition to sort of this other function? And if it isn't, or if it is, then why do you speculate, evolution picked this protein to use as a ubiquitin adapter?
Yeah, that's a really great question. So we know that it can bind histones. If you knock out NAP1L1, you can still make a nucleosome, so it's not required, but there's probably a lot of redundancy. So it probably is a histone chaperone, but how important it is in actually assembling, nucleosomes, I'm not sure.
And follow up to that, I guess, because we are in a genomics institute. I'm curious if it's known if any of these, like the ELACs or NAPL1, are there any mutations known in any of them that are associated with any kind of pathology?
Disease, yeah. So very weakly, it's not like a mutation is linked to genetic disease. It's kind of like, oh, these levels of these proteins is correlated with heart disease or cancers, right? Very vague. So.
Interesting, potentially.
Yes. Cool. Questions?
Oh, yes.
For the NAP1L1 UBE2O interaction, so are they always found to get, or is there some sort of signaling that causes NAP1L1 to bind to the UBE2O at certain conditions?
We overexpress UB2O. It doesn't come down to equal amounts of NAP1, even though NAP1 is very abundant. So that suggests that it's not always there. But if we co-overexpress them, we can purify, if we do a double pull down, I would say that 50% is associated. So my best guess is that they are very good binders together, but it's you have to balance the levels essentially. NAP1 L1 also is palmitoylated, so it might be at other places also. So
maybe adding on to that, do you believe that there are other chaperones that bind to UB2O that might modulate its quality control function?
yeah, so we think that there are other NAP1s that bind, and then there's actually this other that identified, I think it was a viral protein that can bind to UV2 and then target it to certain proteins for degradation. So yeah, I think it's really exciting.
Thank you.
Questions? I think we're good. Susan, thank you so much. This was so exciting. Great to have you share your work with us, and thank you so much for coming again.